So let's get started. So the central dogma, we're going to mention that in a minute, and probably I should have pulled that piece up ahead of this slide. This is a portion of a bigger PowerPoint. Begins with one process, and that process is called transcription. And the word transcribe literally means to rewrite. So, before we had copy machines, there were scribes. They just rewrote books so that they could withstand the test of time. Okay, so a lot of our religious books or documents, they have been rewritten over the years in order to be passed on through the generations, right? So they were transcribed. So that's all that word literally means. And what we're doing is we're rewriting the genetic code into a form that can leave the nucleus, because the nucleus is just your genetic piece, right? We need to get it out into the organelles in order to do stuff with that information. So remember, DNA is made up of smaller segments called genes. Each of these genes code for a specific trait, like your hair color, your eye color, whether or not you can break down lactose. So we need to be able to identify the function of a protein, because genes code for proteins. We need to be able to compare and contrast DNA and RNA, both structurally and functionally. We need to describe the central dogma, big picture, and then go into more detail about transcription and how that happens. How do we turn DNA into an RNA form? There are 3 different types of RNAs that we'll make. So we need to know the 3 different RNAs and what they do. Okay, any questions so far? So when I mentioned central dogma, this is what I'm referring to. I'm referring to getting that genetic code out of DNA, that gene, transcribing it into RNA, and then taking that RNA out into the cytoplasm where it can make proteins at the ribosomes. So this whole theme is the central dogma. We know that proteins serve as the basic structural building block of all cells, and we know that they make up enzymes, and enzymes take part in pretty much all of our chemical reactions that occur inside of a body, right, or inside of a cell. So those enzymes are pretty important, and those proteins serve several roles. So they're formed by the conversion of DNA to RNA to proteins. That right there, that flow of genetic information is called the central dogma. So looking at the central dogma, here's the first process, right? We're going to make DNA, and we're going to make more copies so that every cell has the same DNA. Then we're going to transcribe that DNA into RNA. DNA cannot leave the nucleus, but RNA can. So RNA goes out into the cytoplasm to the ribosomes. If you remember from chapter 7, the ribosomes are the protein manufacturers, the protein factories of the cell. So at that point, we string together a bunch of amino acids to make a protein. Okay, so DNA contains the instructions, and then mRNA serves as a messenger, takes the information from the nucleus to the ribosome to make proteins. So this is the first piece, the comparison. How is DNA and RNA alike? How are they different? So they're both nucleic acids, meaning they're both made up of nucleotides, right? Remember a nucleotide, you guys made models. There was a phosphate, a sugar, and a nitrogen base. So they still have those 3 basic components. They have a different sugar though. So deoxyribose is found in DNA, and ribose is found in RNA. So it's just a little slight difference. One oxygen is removed. So deoxy is this one with the oxygen removed. Ribose contains that extra oxygen. You don't really need to know that, just explaining how they're different. The bases, so remember sugar, phosphate, nitrogen base. They still have adenine, cytosine, and guanine. Both have cytosine, guanine, and adenine. But thymine is no longer available in the language of RNA. So thymine has been replaced by uracil. So anywhere you typically would use thymine, instead of thymine, you're going to use uracil. So this is a pyrimidine. So adenine used to bind to thymine. Adenine now binds to uracil. Cytosine continues to bind with guanine. So the three types of RNA, you have messenger RNA, and its name should tell you what it does. It's a messenger. It takes the message out of the nucleus, kind of like if you were to take a recipe book from the library and you copied down, transcribed a recipe, and then you took that recipe card to your kitchen. You were the messenger, right? So messenger RNA would be like that recipe card, whereas DNA would be the whole recipe book. Ribosomal RNA and transfer RNA. So ribosomal RNA, its name tells you it's part of the ribosome, and transfer RNA says it's going to transfer something. So it's picking something up and moving it to another location. We'll tell you about each of those here in a minute. We got a short video comparing DNA and RNA.
DNA and RNA are both nucleic acids and are made of nucleotides. Nucleotides have a phosphate, a sugar, and a base. But the sugars in DNA and RNA are different. DNA is made with deoxyribose, and RNA is made with ribose sugar. This is where they get their names, deoxyribonucleic acid and ribonucleic acid. DNA and RNA also look rather different. DNA is double-stranded, but RNA is only single-stranded. And for the bases, they both use cytosine, guanine, and adenine, but RNA will use uracil, and DNA will use thymine.
When I was making my comparison, I failed to mention the single strand versus double strand, right? So RNA has a single strand. Maybe that's why it can get out of the nucleus, whereas DNA is double-stranded, so it's bigger. So it probably doesn't fit through the pores of the nuclear membrane, and it can't get out, right? So that is an important difference, single strand versus double strand.
So let's add those. We have our structural descriptions and the 4 different bases. In DNA, adenine forms 2 hydrogen bonds with thymine, and cytosine forms 3 hydrogen bonds with guanine. In RNA, adenine forms 2 hydrogen bonds with uracil, and cytosine and guanine still form 3. DNA is also actually incredibly long, but RNA is comparatively short. There are also 3 different types of RNA. There's messenger RNA, which is used to make a copy of DNA and then can go out of the nucleus. There's ribosomal RNA, which makes up a structural component of ribosomes. And there's transfer RNA, which will bring the correct amino acid in sequence when creating proteins. With RNA, proteins can be built using the code from genes. Genes are segments of DNA that code for a hereditary trait. The section of DNA is copied by mRNA, then built with ribosomes and tRNA in the cytoplasm.
Okay, so DNA contains all of the genes. Messenger RNA will only have one of the genes. It's whatever gene you want to make. That Venn diagram, that would be a great image for you to grab, screen grab, and put it on your study sheet. Okay, so messenger RNA, you got the idea. It is a messenger. It carries a message. It's formed from complementary DNA, which means, remember, A bonds to U now, and C bonds to G. So if I gave you AAA, it would bond with UUU, right? So you want to know the messenger takes that genetic code out of the nucleus and goes to the ribosome so that it can direct that protein synthesis. So that carries the message for one gene. Ribosomal RNA, so ribosomes are actually made of 2 subunits here. And then this is our, so ribosomal RNA is actually part of its component. So it ensures that when the messenger, this is the messenger RNA, when the messenger comes to the ribosome, it'll line up in the right location. Because of that complementary base pairing, it's easy to find its location. So it directs the formation of the ribosome. These 2 pieces will come together when it's time to make a protein. Transfer RNA, what are they transferring? They're transferring amino acids. So it's really important that you keep messenger and transfer straight. The messenger carries the code, transfer carries the amino acids, the actual building blocks, okay? So this is messenger RNA up here. You can see the letter strands, right? We're gonna read it 3 letters at a time. The tRNA comes in a cloverleaf form, and see here at the end, carries a specific amino acid. You have 20 different amino acids. So each tRNA will carry a specific amino acid, and they know which amino acid they need by reading the message. So transcription, notice I have 2 strands of DNA and I'm only going to copy 1 strand. I don't copy both strands. So I want to make a copy of the code, right? Which means I'm going to read the opposite. This is the opposite one is called the anti-code or anti-sense or the non-coding strand. Any of those words you'll see. Since the coding strand is complementary to the template strand, this one I'm copying, since the coding strand is complementary to the template strand, and RNA is also complementary to the template strand, RNA will be the same as the code. So that's a little bit tricky. You kind of often think that the one you're copying is the code. So transcription, you're rewriting the genetic code into a form that can leave the nucleus and enter the cytoplasm where it heads for the ribosome. So transcription itself occurs in the nucleus. DNA cannot leave the nucleus. DNA strand is unzipped, and then new RNA nucleotides are going to be added together to make that RNA strand. The enzyme responsible, RNA polymerase. Remember, enzymes tell you what they do or act upon. This is making RNA polymers. It's complementary to the DNA. When you're done, you'll have a pre-mRNA. It's not quite finished. We have to make some alterations to it. This pre-mRNA will then leave the nucleus where it is then processed. Okay, so just a little repetition here. 1 strand serves as a template. The one you're reading is the template. The other is the coding strand. Okay, so the template or antisense is complementary to the RNA. So RNA polymerase not only adds the new nucleotides, but it's also going to unzip and rezip the DNA when it's done. So mRNA is going to be the red strand here. Notice I'm starting from the 5' and I'm adding free to the 3, right? So I'm creating 5' to 3', which means I'm reading 3' to 5'. Remember, the polymerase can only add at the 3' end, so always add free to the 3. I'm building 5 to 3. So here's where we're at.
How is it that a single cell containing a specific set of genetic material will result in the development of a fish, or a cat, or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the 2 strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene. And after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome.
Okay, so I'm kind of simplifying this for you. When you take AP Biology, we'll go into a lot more detail. They talked about promoters and transcription factors. We're going to talk about those when you take AP. We talked about the 3 different types of mRNA. So one of the things that's different is different polymerase will bond to different promoters. So each type of mRNA has a specific promoter, which is a sequence. So maybe it's TATTA, okay? So RNA polymerase looks for TATTA, and it will bond there, and then it will transcribe messenger RNA. So that's the promoter, the sequence that the RNA polymerase is looking for. And then if you're making tRNA, there's a different promoter sequence. And if you're making messenger RNA, it's another promoter sequence. So that's one of the extra details. And I'm not separating it into initiation, elongation, and termination. We're just kind of like big picture what's happening, okay? So basically you have the RNA now that needs processed. So how do we process it? How do we change it so that all we have is the genetic information that we need? So you have the pre-mRNA and you want to make mature mRNA. So first of all, our DNA has some sections that are coding and some sections that are non-coding. Okay, so exons versus introns. It's really important that you know the exons are the coding regions and the introns are non-coding regions. So the introns may serve other purposes, like maybe they serve for transcription factors. Maybe they are just to separate the genes because you have enzymes that can attack DNA. So if if we add in some non-coding pieces and it's attacked, then that's not going to affect your genes. Introns are unique between you and I. So when you read about or hear about DNA fingerprinting, what they're looking at is introns. Coding regions code for important things, right? So it would make sense that you and I have the same code for things that we both need to do. We both need to make energy. So the code for our mitochondria, the code for the enzymes that are involved in cellular respiration, it's going to be the same. So when we look to identify that forensics piece, we're looking at the non-coding regions because that's the piece that's unique between individuals, okay? We need to get rid of the introns when we make a gene, okay? So we're going to cut out introns and we're going to keep only exons. We're going to add a 5' cap. So remember the 5' end and the 3' end. So the 5' cap is going to help direct that mRNA to find the ribosome and connect at the right spot. So we add a cap to that, and then we're going to add a poly A tail. So a bunch of adenines added to the end. Again, this protects the gene itself from the enzymes in the cytoplasm that might attack the DNA. So this is what that kind of looks like in a simplistic graphic. The blue is the pieces that together make a gene. And the red are the pieces that are just miscellaneous information. So we're going to cut out all of the reds. We're going to splice the introns. And we're going to add the exons together. So you can see the introns are going to be cut out. And all we have are the exons. We're going to add this 5' cap to the 5' end. Remember, we numbered carbons around the circle. The 5' end helps it find the ribosome. The 3' end, we add this poly A, meaning many adenines, to protect it from your cytoplasm's enzymes, endonucleases. That's what they would be called in bacteria. I don't know that they're called that in eukaryotic cells. Okay, so now that we have gone through the whole process, let's take just 5 minutes if we need 5. Identify the function of proteins. What do they do for us? Compare, contrast DNA and RNA. And a lot of times people give me one does this and one does that, or one has this and one has that. Be very specific when you're doing comparisons. Which one has this? Which one has that? Describe the central dogma. Transcribe, rewrite this code. Remember, DNA is going to be complementary to RNA. And then what are the 3 RNAs and their function? On your mark, get set, go. So I saw a few different versions of identifying the protein's function. So structure is a really big one. Transporting molecules, that was a good one. Thinking of our pumps and our carrier proteins. Messages between cells. So I can see you did a little bit of research to add on to this answer. So you're thinking in that case, like hormones might be made of proteins. So for me, just from our PowerPoint, all we had was like a structural component of cells and that they make up enzymes, which then serve purposes. So if you have at least that much, that's great. Structure. So here, Lucy was nice and specific, which has which, right? Instead of saying they differ in their sugars or they differ in their nucleotides. So RNA contains ribose, DNA contains deoxyribose. RNA is a single strand, DNA is a double strand. RNA uses uracil and DNA uses thymine. Excellent. So she listed a bunch of contrasts. Some of you, I saw that you had similarities. So the comparisons. So you identified that they both contained adenine, cytosine, and guanine. Function, so the difference in the function is carrying versus transporting. So, or I should say storing instead of carrying. So DNA stores genetic information, and then RNA carries the RNA or the information. RNA is responsible for protein synthesis. And then DNA is responsible for storing your genetic information. The process of central dogma, this is great. This is as simple as it needs to be. Taking DNA, making RNA, taking RNA, making proteins. To transcribe DNA, she didn't give me the exact copy back, right? Because they have to be complementary. So A bonds to U, T bonds to A, T bonds to A, G still bonds to C, and C still bonds to G. So those haven't changed. So you have U, A, U, G, C, U. When identifying the 3 different types, some individuals gave me functions of the types and some other ones that I've already looked at from other students that aren't in here today. But we wanted to identify each one, right? So the messenger carries the information. Ribosomal directs the ribosomes, and transfer, what is it transferring? Specifically amino acids. Okay, do you guys have any questions on any of these? Want me to repeat any of those so that you can fill that page in? So then the next thing you're going to do, and you'll do this on your own, is just draw out the process of RNA or of transcription. So you need to start with a double-stranded DNA, include a polymerase, make a single-stranded mRNA, show how it changes in trans exons, 5' cap, A' or 3' poly A tail, right? Show how it changes, and that is it. Okay, so you can upload your picture into the same assignment with Google Classroom. So transcription translation, today transcription, tomorrow an overview with Bozeman video, and then Friday we'll do the translation piece. What questions do you have?